Hello students, today we will learn how to do section cutting in botany practicals. Now what is going to be section cutting? You must have already learned in anatomy the innermost parts of the plant body. So to study the tissues or the cells which are present inside the plant body, we need to take the section of the particular material which we want to study. Either you are studying the stem or the root, whichever material you are going to study, you have to take a section of that and see the underlying cells and tissues, and also understand how they are arranged and why they are different from one to another. So today in this practical class, we are going to learn the section cutting procedure. Now in any section cutting procedure, first thing is you have to take out a thin section of the material whichever is given to you in the examination. The material may be a dipole stem, a monopole stem, dipole or a monopole. So whichever is the material given, you have to take first a very thin section of it. Then the second step of the section cutting is, after taking the thin section of the given material, you have to stain the material with a stain called as safranin. So this safranin stain will give the stain to all the lignified cells in the plant body. So safranin will help us to recognize the cells and tissues clearly because the cells get stained by that time. Next is, after the second step, you have to mount the stained material with the help of a drop of glycerin on a slide so that you get a slide which is a temporary mounted slide for observation for the given material. So these are the three steps in which section cutting of the given material is done in botany practicals. Now let us see what are the equipment required for the section cutting. So the material required for section cutting are blade. We require a blade for section cutting, and then we require a brush, we require a needle, we require glycerin, we require safranin for stain, we require cover slips. This is cover slip for making a temporary slide after taking the stain. And we require watch glasses. This is watch glass. And this is the given material, which is a monocot plant grass. Now we will do the section cutting. How to go about this section cutting? Here in this beaker we have the stem of the grass. Now we will take a blade and do the sections. Before that, we'll put in a watch glass a little bit of water so that the thin sections which we cut, we will drop them in this water present in the watch glass. Now hold the material in between these two fingers of your hand. With the help of a blade, try to take out thin sections. Very, very thin sections you have to take out and put them in the watch glass which is filled with water. Why do we put the sections in water? Because they don't get dried up and they will not show any air bubbles if you put in water. So you take thin sections of the given material and make sure that you don't cut your fingers. Be very careful while doing the section cutting. Take out as many sections as possible because we don't know which is thin. For our naked eye, we will find all the sections to be very thin, but when we observe them under the microscope, if they are not even and not thin, we cannot see the underlying cells and the tissues. So take out as many sections as possible and place them in the watch glass. So you find all the thin sections which I have taken from the blade in the watch glass filled with water. Now what is the next step? The second step is, put in this another watch glass a little bit of the stain, that is safranin. After putting the stain, transfer these thin sections which you have taken from this watch glass into the watch glass which is containing the saffron. Okay. We are going to now stain the material which we have taken the thin sections of. Put these sections in the stain for about 1 to 2 minutes so that all the cells and tissues will get properly stained. Leave it like that for 1 or 2 minutes. After that, this is a slide. Upon this, you transfer the stained section with the help of a brush. With the help of a brush, you transfer the sections on the slide. And observe the slide in the microscope. If you observe the slide in the microscope, you will know the cells present in it, what type of section has been given. Since we have taken grass plant here, it is a monocot section. So, if you are satisfied with the section, the stain and the thickness, then you have to take the slide from the microscope and then make a temporary mount of it. Now let's see how to make a temporary mount of this slide. For that, upon the section which you have taken, place a cover slip with the help of a needle. You have to make sure that you don't drop the cover slip upon the slide abruptly. With the help of the needle, hold the needle like this on the cover slip, and before putting the cover slip upon the section, you have to mount it and make it into a temporary mount. So, you have to put a drop of glycerin. For making a temporary slide, we use glycerin, whereas for making the permanent slide, we use Canada balsam. So, you put a drop of glycerin upon the section. That's it, right? And then hold the needle in this position and place the cover slip upon the slide very slowly, so that there is no air bubble formed upon it. Once the cover slip is placed upon the slide, you can observe the slide back in the microscope and give this prepared slide for evaluation to the examiner. So this is how you prepare the section cutting of the given material in your botany practicals.